Hello, everyone. It's my pleasure to welcome you today to Komen's virtual MBC Impact Series event. My name is Casey Volpe, and I am manager of MBC initiatives here at Komen. So for those that are new to the Impact Series, the programming for this series is envisioned as a year-long conversation with attendees, presenting new information and speakers each month, topics selected by those living with metastatic breast cancer. Today, our topic is personalized medicine, and we have a featured speaker is Dr. Gordon Mills, and we have a fabulous moderator named Janice Cowden. Before we begin, just a quick thank you to our 2024 MBC Impact Series sponsors. We have AstraZeneca, Gilead, GE Healthcare, and Merck. We also have Walgreens and Daiichi Sankyo. One more housekeeping note. While our speakers are talking, please feel free to post your questions into the Q&A feature on Zoom. We will get to as many as we can, but please remember our speakers are not able to provide individual medical advice, so try to keep your questions as generally applicable as possible. Towards the end of the programming, I'm going to drop a link in the chat. Please take a few seconds and fill it out. This helps us develop and implement programming for you. And so without further ado, I'm going to introduce Ms. Janice Cowden. Diagnosed with stage 4 triple-negative breast cancer in 2016, 5 years after an early stage breast cancer diagnosis, Janice launched into patient advocacy following training through Living Beyond Breast Cancer's Hear My Voice outreach program in 2017. As a peer-to-peer support and research patient advocate, she is passionate about supporting others with MBC, in addition to continually furthering her scientific knowledge base of this disease, treatments, and clinical trials, which she acquires through attending scientific breast cancer conferences and webinars. Janice is involved with several patient-founded and led organizations, including PCDI, GRASP, and Project Life MBC. As a trained peer support volunteer, she is founder of an international online peer support group for patients newly diagnosed. She serves on the board of directors for MetaViver and is an advisory board member for Project Life MBC. She's an individual member of the MBC Alliance as well. As if that all wasn't enough, she also enjoys traveling, reading, outdoor activities, and spending time with family, including her husband, 2 adult children, and 3 grandchildren, who I'm sure she absolutely adores and keeps very busy. Janice, the floor is yours.
Thank you so much, Casey. I'm thrilled to be here today, and I don't think there's much more I need to really say about myself and my history with metastatic breast cancer, but I'm excited about this topic, personalized medicine. Personalized medicine is very important to all stakeholders, patients, clinicians, pharma, everyone, because in the oncology field, it has the potential to improve outcomes, and also we tailor treatment through precision and personalized medicine that accommodates individuals, taking into account their personal characteristics, their treatment wishes and goals, as well as other comorbidities that they may have, other health issues, and medications that they might be taking. So through using genetic, molecular, and clinical information to design more effective targeted therapies, it reduces or has the potential to reduce unnecessary side effects and improve efficacy. With an emphasis on quality of life, it's patient-centered care at its best, and it also has the potential to have better outcomes for all patients with metastatic breast cancer, and that's something that we all want. And I'm really thrilled that we have Dr. Gordon Mills here today to talk to us about personalized medicine. And I'll just do a brief introduction of Dr. Gordon Mills. Dr. Gordon Mills is the Director of Precision Oncology and Smart Trials at Oregon Health Sciences University in Portland, Oregon. His research area involves developing new treatment combinations and identifying biomarkers to predict which patients may benefit most from them. The overarching goal is to perform deep molecular analysis of each patient to let the patient teach the research team what is important. Dr. Mills, so happy to have you here today, and we're looking forward to your talk.
Thank you very much. I'm just going to try and put together the slides. And I think just to start with, the support and activity of our sponsors and advocacy is absolutely incredible for all of the things that we do. Should come on. Okay, you should see my slides and in the right format. Is that correct?
That is correct. Thank you.
Okay, thank you. Okay, so I'm going to talk about targeting adaptive responses in cancer through smart clinical trials. I'm going to start off, as my colleagues indicated to begin with a little bit of how this is influenced by precision oncology. These are my potential conflicts of interest. I want to point out that there are 3 of them that are important. First off, AstraZeneca supports the clinical trial I'm going to talk about, and one of the key sources of biomarkers that I use are from NanoString and patents that I contributed to, but no longer have any funds coming from them, making them available to the community. So what is personalized medicine? Some of this is going to be obvious to all of you, but I promise it will link to the particular areas that I want to focus on today. Unfortunately, for far too long, we have treated patients in a one-size-fits-all manner, trial and error, rather than using personalized medicine that has been defined as the right treatment for the right person at the right time. Now, I want to note, in terms of therapy for cancer patients, it really is the first time. The first time we treat patients is the best opportunity to have the optimal outcome for our patients. And one of our key goals, and you'll hear about this repeatedly, is that we hope that by moving the precision oncology approaches to upfront therapy, we can replace chemotherapy with therapies that are both more effective and more tolerable. And our overall goal here, in terms of what we do, is to deeply characterize what is happening at all levels in the patient, in their own health, in their own genomics, in what's happening in their tumor, to let the patient treat us what is important and inform how we manage all patients. Now, if we think of where we are in precision oncology, without personalized therapy, some patients benefit, some don't. Frankly, even with personalized therapy, we still see this. But the goal is to go from that case where we can't predict who is going to benefit, who is going to have no benefit, and those who have adverse effects, to population of patients that we can separate and are more and more likely to benefit from the therapy that we are giving them. And the key here is to understand what is happening in the patient and the tumor through biomarkers, so that we can benefit patients far better from personalized therapy. Understanding those that are likely to benefit, those that are not likely to benefit, and those who will have toxicity and should not receive a drug. Unfortunately, although we have identified very good markers for precision oncology with targeted therapy, we lack effective biomarkers for forms of immunotherapy, and also our ability to predict toxicity for patients is much less than we would like. So our goal is to develop more and more personalized diagnostics. And the key for all of the work that we are doing, and really changes in precision oncology and immunotherapy, is characterizing what is happening in the patient, in the tumor microenvironment, at DNA, RNA, and protein levels, so that we can predict those patients who are going to benefit, who won't, and will have toxicity. And I want to emphasize that in the past, we have focused very much on DNA and DNA aberrations, mutations, and genomic changes in tumors. However, we have found that that is inadequate to fully understand how we determine who is likely to benefit from therapy. And I'm going to talk a lot today about also looking at changes at the DNA level, the protein level, phenotype of tumor cells, malignant cells, and the tumor environment, including the immune system, as parts of precision oncology. Now, it's important to remember in breast cancer that we have made incredible progress. We have improved outcomes. The biggest one of those includes a protein assay, which is targeting hormonal responsiveness. So it's not just genomic, it's protein. We've also looked at mutations, PI3 kinase pathway mutations, HER2 genomic amplification, all of those improving our outcomes for patients. And we've also done an incredible job of decreasing toxicity with fewer women receiving chemotherapy with tests such as Oncotype DX. So there are marked differences. I do want to show one slide. Now, this is something that is actually very hard to get accurate data on. This is prior to systemic therapy for our cancer patients in targeted therapy. And if we look at the curves over the years, only about half of patients survived for 5 years at that time. And almost all of that was driven by surgery. If we look at these curves and know where we are today, that blue line is for luminal breast cancer. 98% of patients are now expected to have 10 years of disease-free survival. 92% of what used to be the worst disease of all types of breast cancer, HER2 enriched breast cancer, are now expected to have at least a 92% 10-year survival. These are incredible improvements just simply from understanding that these are very different diseases with different targeted therapies needed and different genomic aberrations. However, where are we today? Well, we're really only at the end of the beginning. We're not where we need to be. We are focused almost entirely on tumor intrinsic effects. We have not paid enough attention to the microenvironment. Most of the therapies that we have are monotherapies, and particularly for metastatic disease, we know that we're going to need multidisciplinary therapy and combination therapy. And there are far too many silos. There are tumor immunologists, there are system therapeutics, there's chemotherapy, there's radiation therapy, and we rarely put them together efficiently. So if we look at where we are, targeted therapy, targeting the genomic aberrations that are in tumor cells, we find that even for patients with a biomarker, only subpopulations of patients benefit from monotherapy. And it's usually short-term, with resistance being almost universal. What we are doing today, I'm going to mention some of the emerging events, is that we are going beyond our standard therapeutic targets by targeting what we call the untargetable, with degrading proteins such as the estrogen receptor, molecular glues, and other therapeutic approaches. Immunotherapy is the most exciting newer area of therapy that is arising. And when benefit occurs, it tends to be durable, making it extremely exciting. It has remarkable effects in a number of diseases: leukemia, melanoma, lung cancer, bladder cancer. However, in most diseases, benefit is modest. Indeed, fewer than 20% of patients on average benefit substantively from immunotherapy. We need to do better. We lack effective biomarkers to determine who is going to benefit and also who is going to have toxicity. And let me point out, toxicity from immunotherapy can be significant with autoimmune type diseases, including unexpected death. There are emerging changes that are really exciting from the current targeted therapies that we use for immunotherapy today of blocking immune checkpoints. These include bispecific T cell activators, directly activating T cells, targeting supporting macrophages, CAR T cells, and antibody mediated toxicity that I will mention along the way. So where are we? Well, it's really time to stop thinking about particular types of therapy, but getting to combination therapies that will be effective for metastatic disease. This is going to be needed to fulfill the promise. However, we have a limited ability to predict combinations, particularly combinations of targeted immune therapy. And I'm going to show you an example. This is going to require deep longitudinal change over time, spatial analysis of what is happening in the tumor at the DNA, RNA, protein, and phenotype of the tumor and the immune system, along with what the hosts themselves. Now, I want to add one other major problem that we are faced with. We are now able to generate so much information on each patient and each tumor that physicians and patients can no longer understand. Patients don't come with barcodes that will tell us what to do. Rather, we're going to have to develop algorithms and approaches that will let the incredible amount of data become information, support physicians' decision. Although we've got some hints of doing this for DNA mutations, it needs to encompass protein, immunology, metabolomics, and spatial analysis. We also have physician and patient compliant visualization approaches so that they can understand the data easily and use it rapidly for our patients. One of the major advantages in the field is AI, artificial intelligence, and there are already a number of algorithms that deal with DNA changes, but we are working very hard to add other levels to that analysis so we can truly benefit our patients. The Komen database approach and the Komen research platform is starting to give us what are called foundational models. If we understand what is happening in large enough patients, we are now able to use that information to begin to predict what we should do for an individual patient. And that's called a foundational model learning. Now, extremely optimistic, but will precision oncology benefit all patients? And this remains controversial. In precision oncology, I'm going to contend we're at the end of the beginning with monotherapy. We need to combine all therapies and biomarkers. Many questions remained, and I use the term frequently, precision oncology rather than personalized medicine. It's very difficult to develop approaches that are specific totally and completely to the same individual. Rather, we are moving to stratified medicine with more and more homogeneous patient groups where we will get marked benefit for each individual in that group. Now, when we look at ductal breast cancer, it's at least 8 different subclasses, and that's one of the problems as we start to implement precision oncology. Smaller and smaller groups that become a set of orphan diseases. Indeed, one of the particular types of breast cancer that I work on is so uncommon that we cannot mount clinical trials to test the incredible set of opportunities that we have for these patients. What portion of patients will benefit? Will it benefit patients when all other therapies have failed, or should we really be thinking about moving it to earlier and earlier therapy where there will be greater benefit? There's been enormous questions of whether it will be cost effective and can we afford both the costs of the testing and of the new drugs that are coming down the pipeline that tend to be quite expensive. And I think a question that has been important is how many patients need to benefit from precision oncology before it's considered a success? Is it 1 patient? Is it 10% of patients? Is it 50? Is it 100? That's a question that's going to have to be answered by ethicists rather than by myself. I contend if we benefit anyone, that's good enough. And one that I think is important to all of us is, can we replace chemotherapy with precision oncology approaches? And one of the important emerging areas that is beginning to do this is with ADCs, or antibody drug conjugates, where instead of giving systemic chemotherapy with all of its toxicity, we use antibodies to target specific aberrations in tumor cells in a personalized manner to deliver the drug just to the tumor. Well, what are the next steps then? We've got to be careful. Cancer is a disease of genetic change in malignant cells. Malignant cells influence the tumor microenvironment, including the immune system. And the tumor microenvironment, including the immune system, is tumor supporting when the patients present or they wouldn't present. If the immune system was effective against that tumor, you would not have a tumor. However, targeted therapy has not fulfilled its promise in many cases, as it only targets the genetic changes in malignant cells. Now, it's beginning to look like when it is effective, it not only targets the malignant cells, but does result in an improved immune system, but perhaps not as strongly improved as we would like. Immunotherapy, in most cases, also has not fulfilled its promise, as it targets only the immune component of the tumor microenvironment. And the tumor that the patient has is a combination of a malignant cell and a changed tumor microenvironment. And the contention, and what I want to convince you of for the rest of this talk, is that optimal patient outcome is going to require combined targeting of genetic changes in malignant cells and the immune microenvironment in a coordinate manner. Now, this slide is a little busy. I'm going to try and walk you through it because it sets a very important concept. Tumors change. Malignant cells and the tumor cells in the microenvironment rapidly adapt to our therapy. Now, this is a statement that is almost self-evident. Tumors initially respond and then they progress. They must be adapting to our therapy. And my contention is that if we understand the process by which tumors rapidly become resistant to our therapies, particularly metastatic disease, we will be able to develop approaches that will improve patient outcomes. Now, why can we target tumors at all with chemotherapy and our other therapies? Well, tumors are under endogenous stress. They're stressed. They're growing in a funny manner. They are in the wrong place. They're metastatic. And indeed, that ongoing stress that tumor cells may be why we have therapeutic index with any of the chemotherapy agents that we use today. Now, our cancer therapy is designed to increase the stress that is happening in tumor cells selectively over normal cells. And it does capitalize on that basal level of stress. So if tumors really do adapt to this stress, and they do this by using pathways that normal cells adapt, cancer cells don't design or invent anything new. They adopt other processes in the body. If we can measure and target those adaptive responses, we may have a novel therapeutic opportunity for our patients. We have three different ways that we could do that. The first of those is we could hyperactivate or increase the amount of stress. And we do that by targeting particular pathways. I'll mention them a little later on. You could block the survival pathways that cells use to deal with the stress that they are receiving. And those pathways I'm again going to talk about a little later on. And the other is that stress itself changes the tumor. I just mentioned that. It changes the immune system in the tumor. It changes angiogenesis in the tumor. It rewires the tumor broadly. And that could be a whole set of new therapeutic opportunities that we can capitalize on. Now, I want to emphasize that these changes occur over days to weeks to months. These are not major genetic changes in selection of tumor cells that happen late. These are how the cells and the microenvironment change their states that we need to understand. Now, that is why we developed a program called SMART, serial, meaning looking at what happens over time, how do things change, measurements of molecular and architectural response to therapy. This is beyond genetics. Indeed, if we look at what's happening today targeting genetic aberrations in tumor, only 40% of patients have actively targetable events. If we could target them, if we had the greatest drugs in the world, it still is not all of the tumors that we have in terms of the way we can target. That means we look at more than DNA and genetic events. Now, patients are treated frequently on a pre-therapy biopsy, and sometimes that's many years prior to our therapy. Yet we don't ask how the tumor changed, not just the malignant cells, but the tumor ecosystem changed over time. We have to know that. And our hypothesis is that optimal patient outcomes will accrue from treating patients based on how the tumor and the tumor ecosystem adapts to therapy. There's a corollary, and that is that you have to act on spatial analysis of malignant cells and the tumor ecosystem, including immune cells and communities as they adapt to therapy. And this requires a detailed analysis of DNA, RNA, protein with single cell, looking at individual cells in spatial resolution in real time. And I've listed just some of the assays that we use to do that in this program, and we make those assays available to anybody who wants to use them. Now, I'm going to use a drug as an example, and these are PARP inhibitors that have been already approved for use in patients with breast cancer with aberrations in BRCA1 and BRCA2. And indeed, they came into focus as synthetic lethality with defects in homologous recombination. You can read that as BRCA1 and 2 deficiency. However, what we have found since we've started using these drugs, that they have other effects that are really very important. And they can induce DNA damage that causes marked stress to tumor cells independent of BRCA1 and BRCA2 abnormalities. And they do this in multiple different ways. They block repair of a particular type of DNA damage called base excision repair. They also block the replication fork. And you may remember back to high school, it's hard to replicate DNA, particularly that trailing strand. And so it gives lots of DNA damage. And further, the PARP inhibitors stick to DNA. They keep PARP in place and it doesn't really get released. And so you get more DNA damage. And we can capitalize on that DNA damage. It's much less toxic than classic cytotoxic agents or chemotherapy agents, but we can capitalize on that through combination therapy that are effective in multiple diseases, including breast cancer. This slide perhaps is a bit busy. I will go through some of the points in it. But if we're going to target adaptive responses, there are some key things that are required. The first one is if we're targeting metastatic diseases, the changes that occur on therapeutic challenge have to be the same in different metastatic sites. And indeed, we have tested that extensively and have found that each patient has their own set of adaptive responses that we can measure and capitalize on. They have to be different across different individuals. If everybody had the same response to the therapeutic agents, we'd cure everyone. So the adaptive responses are different. And indeed, I've got a list of those down below in a few minutes. There have to be a limited number of adaptive responses. If there are too many, we couldn't develop therapies or have therapies that would work, and they have to predict combinations that will work. And we've shown that both of those are true in clinical trials. And then we have to ask a very important question, and I'm going to show you some data, is can we predict adaptive responses, or do we have to measure how the tumor changes step by step to find the optimal therapy for each patient? Through many studies in patients, in model systems, we have looked at adaptive responses and resistance responses to PARP inhibitors. And there are indeed a limited number of targetable pathways. They occur across different lesions in an individual patient, and the pathways are targetable. I've listed them here, but you don't need to pay attention to that in any great detail. However, we have been able to develop multiple trials using this knowledge where we have either preempted or measured the changes and taking those forward for patients with breast cancer, ovarian cancer, endometrial cancer, and some other cancers, and have shown remarkable responses for those individuals sufficient that pharmaceutical companies have multiple registration trials of this ilk, either planned or in progress. I want to show you one of those that we're doing today and how deep characterization of the tumor can allow us to find biomarkers that will identify who benefits. Now, before I do that, I want to note that olaparib induces rewiring of the tumor and alters the immune contexture. It increases interferon production, it increases immune responsiveness in BRCA1/2 wild-type patients. And this is going to focus solely on patients with triple-negative breast cancer who have normal BRCA1 and BRCA2. So we're taking that population where we have limited amount of therapeutic options with targeted therapy and immunotherapy. And what we do is we start off by having a biopsy from the patient, and we do real-time deep analysis of DNA, RNA, protein of the tumor, of the ecosystem, and of what's happening in blood. Patients then receive olaparib for 28 days in the studies I'm going to show you. And then we repeat that deep analysis in an on-therapy biopsy. Let me note that this information is going to be used in the treatment of that individual. This is not a pure research program, but rather we can look at every single patient and say, we intend on using the information in your care. And this has been received extremely well by our patients. Indeed, we have yet to have a patient who has said they did not want the second biopsy. We have had a couple of cases where that second biopsy was not successful. First off, the tumor had shrunk so much that the interventional radiologist no longer felt that they should take a biopsy, that it would be dangerous. And then in other cases, when we took a biopsy, there was no measurable tumor to work with. So this is suggesting that even after 28 days, we can have remarkable responses. Now, we do the studies in a two-phase approach. It's called a Bayesian analysis, where we first treat with 15 patients and determine that we are getting a good response, but also use that data to develop or refine biomarkers that will be evaluated in the next 13 patients. So we really get both activity and the development of new biomarkers. Now, I want to show you the results because after that 28-day treatment, the patients continue to receive the PARP inhibitor olaparib and durvalumab that targets the immune system through blocking PD-L1. Now, this is what we see when we look at patients for progression-free survival and overall survival. This is metastatic breast cancer, and I've included in these patients with a progression-free survival on the left-hand side, overall survival on the right-hand side, and then mark the two most recent approvals for active drugs, pembrolizumab, targeting PD-1, PD-L1, and an ADC, an antibody drug conjugate targeting TROP2. And though you do not ever want to compare directly across clinical trials, what I can say is that the responses for these patients is actually as good or better than any that we have seen in ongoing trials for similar populations. However, that's not what we want to do in precision oncology. Rather, if we look at the patients in this trial, this is 23 patients that have received this therapy and close to completing the trial. And what you can see here with months at the bottom, note that's 40 months at the bottom, in patients where chemotherapy is under 10 months of survival time, and those top level therapies are around a year. We have a population of patients who have progressive disease, PD, and we need to do something for them or identify them. And we have a group of patients that have remarkable responses. We need to be able to identify who those patients are. Now, before I go on to do that, I do want to note some things here. 2 of those patients that are still alive have been off therapy for more than 18 months. With triple negative breast cancer with conventional therapy, if you are beyond 6 months off of therapy, the chance that you will recur is under 1%. There's another patient that I can't include as part of this trial because she presented with brain metastases. And unfortunately, in most cases, patients with brain metastases are not allowed to be on our clinical trials. She received a PARP inhibitor and a PD-L1, PD-1 immune inhibitor and had remarkable results despite having brain metastases. Indeed, she's been off therapy for 2 years at her own choice because she was 37 years old and wanted to have children. And soon I'll be able to show you the picture of the beautiful young baby that she has had after being off therapy with a disease that we rarely ever get to talk about these types of responses. So precision oncology requires that we identify those patients with major benefit. It also requires that we have something to do for the patients with limited benefit. And to do this, we are characterizing tumors deeply at the level of DNA, RNA, and protein. And we characterize malignant cells in the microenvironment. By the way, all my slides with data will have the people who did the work down at the bottom. I just get to talk about it and raise the funds so that they can do the studies. Now, I'm not going to show you the details of this list, but I want to point out that we have developed an in-depth set of assays that are both research and available in the clinical environment that allow us to look at what is going on in tumors at a depth and breadth never done before. And indeed, I'm comfortable saying that because I average over 100,000 air miles a year visiting people to see what they are doing. We make these assays available to anyone who wants to use them, either by running them for them or teaching them how to run them. This is one I just want to point out because it is totally unique. In order to use assays for patients, they have to meet CLIA criteria. That's part of what you have to do in the United States. This is the first and only multiplex protein assay that lets us look at 90 different proteins that are critically important to predicting how drugs will work. Talking about PARP inhibitors as an example, all of the pathways are listed there, from immune through signaling pathways through cell death modules. This particular platform drives many of the steps that we can take because not only is it CLIA compliant, but is cost effective and rapid. We can get data back within a week to 10 days. Now, I'm not going to go through this slide again. What it does is shows you all of the different molecular markers that we found to be statistically significant in the first phase of our trial. 32 independent markers able to identify those patients who were likely to benefit from this combination of PARP inhibitors and PD-L1. Now, I want to show you something that is very important, though. Here is an example of those. This is just looking at the immune environment with 3 different assays, 2 of them protein-based and 1 of them RNA-based. And what you see pre-therapy in a Kaplan-Meier plot is that you want to see that you have statistically significant ability to predict who will benefit and who will not. And in that plot, there really is nothing on biopsy 1. P-value is just not predictive. But we ask now how the PARP inhibitor changed the immune environment in that on therapy biopsy. And you can see here that you identify a group of patients in red that do very poorly, and you can identify all those patients who are going to have a marked benefit. So we can now segregate and say, you are likely to benefit, you are not, a very powerful biomarker. I'm going to show you another biomarker, and this is one that I developed with colleagues while I was at the MD Anderson Cancer Center. And it again measures immune activity with a unique set of markers. And we're not entirely clear why it works as well as it does, but it is actually very, very powerful. And again, this one has some utility in the pre-therapy biopsy. If you look at the data here, if you're red, if you have a high BLIS, basal-like immune suppressed signature, you do poorly. If you have a basal-like immune activated signature, you do well. However, on therapy, the data is much more predictive. About 80% of the data can be acquired on that pre-therapy biopsy, but if you want the best predictor for your patient, you need the on-therapy biopsy. If we look again at patients and their survival using that same idea of who is going to benefit or not, this is progression-free survival, so the timeline is shorter than that of overall survival. And what you see here in blue is those patients who have marked benefit, and in red, those who did not. So one of the important things that we've been able to do is to develop that suite of biomarkers that are highly predictive. And we were then able to test them on a completely independent test set and show that 89% of the patients were predicted accurately by our prediction. This is an unheard of number. HER2 testing is only 50%. 50% chance you will benefit or not from standard HER2 targeted therapy. So this may be getting close to being able to really determine who's going to benefit or not from this therapy. Now we have a question though that arises is what will we do for these patients in red who did not benefit? And so we don't want to just treat those patients in blue that benefit, but have options. So one of the neat things about the way in which we do this study is that deep DNA, RNA, and protein analysis allows us to look at what's happening in those patients who didn't benefit and ask, is there emerging opportunity for those individuals? The quintessential survival pathways in cancer cells, and indeed in any cells, is activation of two different pathways, the AKT pathway and the MAP kinase pathway. They allow cells to survive. In particular, the PI3 kinase AKT pathway. They do also allow cells to proliferate. You have to be alive to proliferate. That may be part of the major reasons. But if we look over here at these patients in red and ask whether they had those pathways activated or not, MAP kinase, AKT, both, AKT, AKT, MAP kinase, MAP kinase, MAP kinase. Remarkable that this seems to be the survival pathway that is being used by those patients who do not benefit from our PARP plus PD-L1 inhibitor combination. And indeed, we've seen this in other studies that we have done previously and have tested ways to target those pathways. Now, if they are really important to resistance, they should predict outcome. And here again is one of those Kaplan-Meier plots. If you have high levels of the pathways, you do very poorly in blue, and that's survival in months. And if you have on the right-hand side, that's disease-free, and that's overall survival. So really, in that second biopsy, we have an incredible ability to predict who is going to benefit or not. This has led to our ongoing trial, where we do deep real-time characterization of the patient in the patient tumor. We take a second biopsy at 14 days, so we have time to analyze it. We assess the adaptation that we see happening to the PARP inhibitor. How did the tumor change to allow it to survive the stress from the drug? And then pick the therapy. If they have immune activation, they're going to get that PD-L1 data that I showed you. If they have activation of the RAS-MAP kinase pathway, we have drugs that target that pathway. If they turn on the PI3 kinase AKT pathway, we have drugs that target that pathway. And if just simply the amount of stress goes up from that first set of ways of doing it, we have drugs that increase the amount of stress in ATR and SERD. We don't have enough data from the other three arms yet, as the immune activation arm is the most common. Now, don't want to do biopsies if I can avoid it. Multiple different things from tumor cells go into the bloodstream. And so we can monitor what is happening in tumor cells by monitoring changes in the bloodstream. Now, I want to point out that this is aspirational. We're not quite there yet. We can look at DNA in the bloodstream to ask whether the tumor is responding. We can look for estrogen receptor mutations that change our therapy approaches. We can already tell what type of breast cancer patients have by what we're looking at in the bloodstream, and this is important to accurate therapy. We can look at what's happening to immune activation in the bloodstream. It does predict what's happening in the tumor to a degree. There are cytokines released into the bloodstream that we can measure. There are tumor cells in the bloodstream we can measure. And further, RNA and protein are released into the bloodstream, so we can now look at DNA, RNA, and protein in what we find in the bloodstream. We've not yet been able to deconvolute that sufficiently to change patient management, but it's there. And then all of my slides as we went along to thank those people who did the work. But really, to make something like what I described happen, it takes a village. And these are the people that contribute to the overall program at the Knight Cancer Institute that I hope have helped convince you that we're making major progress for patients, and that understanding how tumors adapt to therapy is going to make a major impact in the future. Thank you.
Thank you, Dr. Mills. That is fascinating research. And from the patient perspective, I just want to say thank you. We appreciate the work that you're doing. And we do have a couple of questions in the chat. So the first one is, you mentioned that immunotherapy use in patients that have autoimmune disorders can lead to unexpected death. Does that mean that a patient with metastatic breast cancer who has autoimmune disorders can or cannot use antibody drug conjugates?
I won't say that you can or cannot, but this is something that we would look at very, very carefully. Further, I think what I said or meant to say is that the drugs that we are using can cause autoimmune disease. And particularly the checkpoint inhibitors that can, we're much better now. But at least some of the initial studies did have cases where that was so significant that patients died. We don't use the worst of those drugs at this point, other than exceptional circumstances, and in essence, not in breast cancer. So I'm sorry if I gave the wrong idea. Now, autoimmune diseases and ADCs, antibody drug conjugates, probably are not linked. It's the checkpoint inhibitors.
Thank you for that clarification. The next question that we have in the chat is asking about studies using the combination therapy of PARP inhibitors and immunotherapy together. Have there been studies done on metastatic breast cancer patients who are not triple negative? And if so, this is a two-part question. Have there been any lobular breast cancer patients included in these studies?
Okay, well, I showed you already our study, which is using PARP inhibitors and immune modification, and that has been reported and is really receiving strong support to move on to a next phase study. There have been other studies that are similar, although small, and it's the responses to some of them have not been as exciting as we would like, and we're beginning to understand why. Now, I do not know that any studies with lobular breast cancer have been reported. I won't say that there aren't studies in progress or that lobular breast cancer patients may not be included in some of the studies, but none of them, to my knowledge, have yet been reported.
Thank you. We have another question, and this one is, what barriers are there to personalizing medicine for an individual?
So there are many. I mentioned a few. The first one is we don't have drugs for all of the abnormalities that we find. Secondly, I think the most important of those is the one that I tried to emphasize, is that we've not been able to figure out how to use combination therapy for most of those. There is the cost, both the cost of testing, which turns out that all of the testing that we do in the CLIA environment is less than 2 CAT scans. And CAT scans are done routinely. So I don't think that is a major problem. But I will tell you that many of the drugs that are now being used are extremely expensive, between $15,000 and $30,000 a month. And this precludes their use in individuals who don't have high-level insurance, and certainly in emerging countries. And that is something that needs to be dealt with. So if we talk about the easy ones, and I'm going to say easy here and almost tongue-in-cheek, right drugs, right assays, clinical trials, and then community support for how we're going to deal with the costs. And that's the hard one.
Absolutely. You know, what you mentioned, financial toxicity is certainly a huge issue in the metastatic breast cancer community because most patients are on treatment for life. And as a patient who's engaged and involved in that community, I see it all the time. And it really is something that I hope will change in the future. Okay, I have a couple more questions here. The next one would be, what does the future of breast cancer research hold? What's coming soon that you believe will have an enormous impact that will benefit the patient most?
Okay, so there really are a number of exciting events, and I mentioned some of these along the way. First one is we have whole new concepts about developing drugs that target the abnormalities that are present in tumor cells. Instead of focusing on kinases or enzymes that we were able to target, PARP inhibitors and the PARP enzyme, we're now able to actually degrade proteins. And that brings in a whole set of targets we hadn't been able to target before. We have molecular glues, we have other approaches. AI is vastly improving our ability to move those things forward. We now have programs that will give us the structure of every known protein with reasonable ideas and give us an idea of how to target them. So those are massive changes that are going to revolutionize what we are doing. The other one that I think is just as important is our ability to work with the immune system. So far, the targeted therapies that we've had in solid tumors and breast cancer have been solely, proved at least, targeting PD-1, PD-L1, and that's it. There are many other immune checkpoints, so CTLA-4, others that we can target in the appropriate patient if we had the right biomarkers. So that's step one. Secondly, there are now ways to directly activate T cells by cross-linking receptors on their surface. And the only way we've found to do this is with bispecific antibodies. Now, this is wild. You build an antibody, it binds one protein, but it's got 2 strands. What we do is synthetically make one that has one strand that binds one protein, one strand that binds another protein, and lo and behold, you bring the 2 together, and that's how things get activated. And this is really looking exciting in early studies. CAR T cells, where we actually take T cells out and NK cells in some studies, and then genetically manipulate them so that they will target the immune system, so that they will persist, has just been approved for the first time in solid tumors, not in breast cancer. Similarly, something that I played a role in over 40 years ago, which is we can now activate 2 T cells outside of the body and then give them back, and that has finally been approved. And it does take that long in a disease, not breast, but it'll be applied. Now, the one that is perhaps most exciting right now is the idea of antibody-drug conjugates. There are different proteins on the surface of tumor cells. Most exciting one right now is HER2. Targeted with drugs previously, but right now we're using it because it's on the surface of tumor cells. And by the way, not just HER2 amplified tumors. Most breast cancers have some HER2. And even HER2 ultra low applies to what I'm about to say. is that HER2, when bound by an antibody, will internalize. That's how it works. And if you take that antibody and put something on the end of it that is toxic to the tumor cell, that becomes an antibody drug conjugate. And there are two classes of those, and they are looking spectacular. It's changing how we think. Now, there are other targets that are relatively specific to tumor cells, and a massive effort is happening because of the success of things like targeting TROP2 that I mentioned earlier and HER2. Those are probably the most imminent. Now, I'm going to contend that the real excitement is when we start to combine therapies. And we need to remember that radiation therapy, chemotherapy are highly effective. We shouldn't discard them. Love to get rid of them if we can, but they still are effective. And targeted therapy and immunotherapy is effective. So figuring out how to use these together without toxicity is really, to me, the most exciting next step.
Absolutely. Thank you. It's very important that we get that right drug to the right patient at the right time, but also at the right dose, and always try to preserve quality of life. I have another question here for you, and it's regarding the SMART study. What phase is it in currently? How much longer do you think it will be before this SMART study will be broadly applicable in the clinical setting?
So it's phase 2. And we've just submitted a paper for the world to review and see what they think about it. And it's just on the first arm. So we've got 3 more arms to complete. That's going to take a while. It's going to take some funding challenges and some other things to work out. But the first report is under review right now. We expect to complete both phase 1 and phase 2 of that first arm imminently. So that'll be the second major report. We are negotiating a follow-on registration study or registration type study. I'm not sure it'll quite be registration, using the biomarkers that we have discovered to look at combinations of a brand new PARP inhibitor that is both less toxic than olaparib and more specific and better at combinations, plus one of those bispecific antibodies that will activate the immune system with the hope that we can move those forward. So that's under discussion and negotiation with a number of companies because of the power of the biomarkers that I showed you.
Oh, that's excellent. You know, for patients, research can't happen fast enough, but we do know that it always takes years to get from that experimental drug to getting to the clinic. So we're really excited about this. I did have one quick question from our Facebook group, and I'm not sure if it's something that you can address or not, but I'm going to throw it out there. This is actually about having dense breasts. And if there is someone that has dense breasts and breast cancer runs in the family, do you believe that another test would be helpful other than a 3D mammogram?
Well, there's a whole series of answers to that, and others might be better off, but I can give you some. First off, if cancer runs in the family, genetic testing is the first thing to do, and that could help inform how to do testing. Secondly, today, MRI and the tests that we have are the best we've got. However, that population of individuals will be looked at carefully because of the challenges and the incidence of breast cancer with emerging approaches of early detection. So the answer is today, that's the best. Family history, get your testing done. Actually, a study just came out of Canada and where I'm from, you may or may not recognize my accent, that says testing all women for BRCA1/2 will be both cost-effective and improve outcomes. So that's a critical study that needed to be done. We're working on a population-based study in Oregon to try and test that concept. Genetic testing is step 1. Continue the screening which is the best that we have today. And we need better screens, and we're working on them.
Absolutely. Well, unfortunately, we are at time. And I just wanted to say thank you so much for coming here today and providing all of this excellent information for us. And it's really appreciated. And at this time, I'm going to give the microphone back to Casey.
Thanks, Janice. All right, guys, thank you so much to Dr. Mills and Janice Cowden for being a fabulous moderator, a fantastic and fascinating presentation. The audience hung on there in there with us, even though we're a little past time right now. So I'm so appreciative. Hey guys, today's session was recorded. It's going to be posted to our landing page soon, and it'll come out. If you register for today's event, it'll come right to your email as well. But please go ahead and visit Komen.org/MBC series, and you can catch other sessions and other topics you might have missed that are valuable or interesting to you. Once again, I have to take a moment and acknowledge our sponsors, because without them, we wouldn't be able to bring these fascinating presentations to you. So thank you to GE Healthcare, AstraZeneca, Merck, Gilead, Walgreens, and Daiichi Sankyo. Last but certainly not least, there is a link in the chat with to a survey. This helps us determine topics. It helps us get feedback on how we're doing, how the events are structured, what topics we cover. It's really important. It doesn't go into the mystery abyss. I get those responses, and I design the program accordingly. So please take a moment and fill that out. It helps me out a great deal. Finally, last but not least, our next event won't be until November, and it will be focused on how to manage side effects, specifically after being in treatment for a long time. So stay tuned, check it on our landing page when that information is live, and we've got it formally scheduled so you can hop on if that's interesting to you as well. So thank you again for joining us today. Thanks to everyone who made it possible. Stay safe and stay connected.